this summer, my lab and I have been trying to model, which means to create a treatable system of a disease called hypodiploid acute lymphoblastic leukemia, or hypodiploid ALL. And ALL is the most common childhood cancer, and we have been trying to create a subset of ALL that the clinic sees commonly, but is very poorly treated just due to its therapy resistance. And what differs ALL and hypodiploid ALL is regular cells will have 46 chromosomes because that's just one set of 23 from each parent, but hypodiploid ALL likes to cut up a bunch of the chromosomes and spit them out. And then they'll divide rapidly and without any without any response therapy because they have so few chromosomes present. And we see a lot of mutations on these chromosomes that remain, and those mutations can create critical growing spots for like a lack of differentiation, so they don't become mature B cells and they get stuck, and then they grow really, really fast and make tumors and flood the blood and bone marrow so that everything gets bad is what happens. So what we've been trying to do is eventually make a mouse model of this cancer, and we've been using a technique called CRISPR-Cas9 to imitate this cancer by knocking out quite a few genes that are seen in this cancer as well. So we have been trying to edit out genes called TP53, NF1, and then a few B cell genes. These two are very common tumor suppressors. TP53 is responsible for LFS. And here you can see that these proteins are all quite well expressed, and then you look at IKZF2, and IKZF2 is very, very light, it's not very dense, there's not much going on there. And then same with IKZF3, and then same with NF1 here, there's actually nothing as we can tell. And then same with IKZF2 again for the different cell line. So this is the result of protein expression after we use the CRISPR-Cas9 editing techniques. And we infected these cells with the CRISPR complexes, and we ran this test to see how much protein they still express. And these genes have been deleted to the extent where they can't actually produce functional protein anymore. So with this being done in cell lines, so these are cells you've grown in a dish, we've been expanding these techniques to edit human stem cells and cord blood that we've received from patients who have just given birth. So we took this cord blood from their umbilical cords, and we have been editing these stem cells that these cells can actually create every single blood cell in your body. They create the platelets, they create red blood cells, they create white blood cells. So we've been editing these with the same techniques here to knock out these 4 genes, and eventually we're going to sequence the DNA and see if the mutations are present. Because we don't have enough cells to do a Western blot like this, and we also don't want to use these cells because these are expensive and valuable. These we can grow as many as we want all the time, so we're going to use this technique to make sure the mutation is present, and then once we do that, we're going to plant them into mice. And these mice are bred specifically to have no white blood cells. So these human blood stem cells will reconstitute or replace all of the white blood cells in the mouse. So instead of treating a mouse with cancer cells, we'll be treating a mouse with human cancer cells. And that makes the treatment even more relatable to a clinic and hopefully more effective once we start experimenting with clinical drugs. That's what we've done. So basically, we looked at a cancer called hypodiploid ALL that has very low outcomes, very therapy resistant, and we have been trying to model it in a mouse so we can eventually treat it in order to increase those survival rates for patients diagnosed with this cancer.
I have a question.
Yes.
Why a mouse?
Why a mouse?